Now, okay, so just to get back to it here, we have the fatty acid linked to the glycerin backbone, and this is again one of those kind of condensation reactions where we're going to make a molecule of water and we make a carbon-oxygen link. And what we can do is take one of the end fatty acids, not the middle, and add a phosphate group to it. And that puts that negative charge there, which makes this molecule somewhat schizophrenic in that it can interact with water with the phosphate end and interact with lipid and oily stuff on the triglyceride end. And so, as biomolecules go, it's a pretty critical thing for making the lipid bilayer. All right, I think this is where we finished up. And let's look at this, and we actually go to the board here for a moment. Because this actually is an important part of the biochemistry that we'll be talking about, particularly when we start talking about metabolism things and enzymes. But proteins. These are made up of amino acids, and as we say, we got 20 different amino acids. And these are the monomers. What makes proteins kind of interesting is that both carbohydrates and lipids don't have much in the way of charge differences. But the carbohydrate is just, for most of them, glucose or at least some OH-filled sugar molecule. Just that's all there is, just sugar. And so it's got a real strong negative charge, but it doesn't really do a whole lot of interesting structures and things. With fatty acids, yeah, we can make a phospholipid that's got a charge in it, but again, it's just a really long stretch of nonpolar molecules. Now we're getting into something in which we actually have some variety in this part that we call the R group. And so what we're going to do is begin linking together amino acids and create what's called a peptide bond. And when we have it here, it's actually an important thing that I want to spend a little bit of time on. So Victoria, you're getting a one lecture ahead. So it's spoiled. So when we think about amino acids, we have, for at least our purposes at this point, 20 different ones. Now, the cool thing about most bacteria is they have the ability to biosynthesize every single amino acid. You and I cannot. We can biosynthesize 13 of them, but there are 7 amino acids that we can't biosynthesize. Consequently, we have to get them from other organisms, or I guess from, you could eat people, but you got to get it from an external source. Whereas bacteria, if you feed them a nitrogen source and you feed them a carbon source, while they will take a long time to grow, a lot of different wild-type bacteria are absolutely able to make every single amino acid, and they can grow on the most primitive kinds of, quote, food, stuff that we absolutely could not survive on. So that's a pretty cool thing. Now, the amino acids have the R group. And that R group, we can categorize it by, if you take my biochemistry, you have to memorize all the R groups, but fortunately we don't need to at all. We can categorize them based on their chemistry. We have R groups that are polar, and some have a positive charge, some have a negative charge. We have R groups that are nonpolar. We have some that are acting as acids and some that are acting as bases. So we can talk about acidic amino acids or nonpolar amino acids. And what this means is there's a lot of charge heterogeneity. It ain't all just straight negative charge. And what really makes proteins sing is when we begin linking them into a polymer. And let's look at what happens here. We can develop a hierarchical set of structures that we refer to as the primary, or as one with a degree, but that is the primary structure. the primary structure is critical, and the definition anyway is pretty easy. It is simply the amino acid sequence. And as a molecular biologist, what I like about the amino acid sequence is I can deduce this from the nucleic acid sequence. So if you hand me the DNA sequence of an organism, I can give you the proteins that organism can produce. And with bacteria, it's really pretty easy to do. So by knowing the primary structure, that means we know the amino acid sequence. Now, this is held together, of course, by the peptide bond, by a covalent bond. So when we start thinking about some of the enzymatics and biochemistry, let's keep in mind that it takes a fair amount of energy to break a covalent bond. We're not going to break a covalent bond just trivially. These are the tough guys. We've got to set them on fire or use some kind of enzymatic magic. Now, the secondary structure is defined as local R group interactions. And local meaning probably, who are the amino acids in your neighborhood, 5 amino acids on either side. So that's the local stuff. Remember, proteins can be upwards of 1,000 amino acids. And now, when we look at the bonds that are responsible for this, it's things like ionic bonds, hydrogen bonds, various sort of charge interactions, and polar, non-polar interactions. And just a real standard example would be a stretch of like charged amino acids. So we've got and we're going to put a negatively charged R group here in a stretch. And we'll put a positively charged R group stretch over here. And then just for the heck of it, put a couple of non-polar guys here. All right. These like charges, as we like charges are going to actually repel each other. And what happens is the R groups with their like charges are beating on each other, and they begin to actually twist around the central carbon such that we end up actually having the charge distributed in sort of a twisty fashion, and that they spin around to give each other room. This manifests itself actually as a helix. And so you're going to have this protein develop this kind of shape where you have the nonpolar stretch in the middle. And this is one of the standard kinds of secondary structures known as an alpha helix. Another standard secondary structure is a zigzag in which approximately every fifth amino acid can hydrogen bond with the guy above it. And you make this structure that's known as the beta pleated sheet. And now if we look at the interactions that hold these guys together, we got charge interactions and hydrogen bonds. What I think of in my own official biochemistry terminology, what we refer to as them weak-ass bonds. So primary structure is held together by tough bonds. Secondary structure is held together by weak-ass bonds, meaning that really slight changes in conditions can break these bonds. You change the salt concentration just a little bit. You change the pH just a little bit. You change the temperature just a little bit, and you can begin breaking these weak-ass bonds. So these are bonds that are very easily manipulated. Now, lastly, the third structure, you guessed it, is known as the tertiary structure. So tertiary structure, I don't know if you can see that. Yeah, close enough. Tertiary structure we define as now, it is essentially non-local, non-neighboring R group interactions. And that means we're going to fold the protein up. So we have something that might look like this, where we've got the negative charges on one side and the positive charges on the other side interacting. And you begin to fold this protein up. So this becomes the tertiary structure. And I will tell you that it can be a little hard to predict the tertiary structure, and actually the secondary structure too. It comes down to the thermodynamics of the protein structure. Proteins are going to fold up into the most stable and least thermodynamically requiring structure. So you're not going to have things that stretch way, way across and things if they can just sort of tuck themselves in. What this means is that we can make a pretty good guess about a protein's secondary and tertiary structure, but probably there is not one single perfect tertiary structure or there's actually a set of them that are about the same. And for the most parts, in terms of protein work, the critical pieces are probably all in the right shape, and there's just little tiny folds and changes. But this actually becomes a major part of the production of vaccines and all sorts of stuff, in that the proteins in their native structure are what the immune system recognizes. And so it's important that we have the appropriate structures being formed here. Now, again, with tertiary, these are, as we would say, the weak-ass bonds. So when we start doing things like, running a slight fever, what you're doing is disrupting the secondary and tertiary structure. And in fact, as we'll start seeing, we start talking about ways of sterilizing stuff. We can do things that'll disrupt the secondary and tertiary structure. And one of the points we're going to be getting into here is that proteins work because they fold up into their secondary and tertiary structure. When we remove secondary and tertiary structure, we can call this the lame-ass marker. The loss of secondary and tertiary structure is referred to as denaturation. As it turns out, every kind of process you can think of to cook stuff or to sterilize stuff is going to be involving protein denaturation. Actually, as I say sterilizing, it's not entirely true. We can do things like radiation and UV light, which is shattering the DNA, and it's preventing the organism from carrying forward any further metabolism or being able to divide. The proteins themselves can still be intact, which actually is an interesting point, is that you can have something that's actually sterile, but it actually can be fairly nasty, dirty. And for some molecules, there could still be some toxin things floating around, even though there is no life there. It's sterile. And so we can make a distinction between something being sterile and something being sanitary. Things that are sanitary, they clean. Things that are sterile, they just don't have any life. There's nothing that living in them. So you can take mud outside, sterilize it, but I ain't going to recommend eating it. You probably could, but no, it may be bad for you. Don't do that. Anyway, so when we start to heat things up change the salt concentration, change the pH, what we're doing is changing the structure of the protein, and we risk denaturation. So, oftentimes, when we denature a protein, it falls out of solution. And so a classic example of this is heating an egg. The white is almost pure protein and water, and when you heat it up, the protein falls out of solution. And some proteins, when they fall out of solution, you actually can bring them back into solution. And there are a whole bunch of pharmacological agents, proteins, that are precipitated out and vacuum freeze-dried, and you can resuspend them. You just shoot a little bit of water in, shake it up, the protein refolds and goes right back to life. And so you get lots of proteins that will renature, but there are a whole other bunch of them that once they denature, man, they are never going back into solution. And the major protein in milk, casein, does that as well. So you drop casein out of solution by making like cottage cheese. You can't get it to go back into solution. You drop egg white out of solution. Egg white's like a classic one. It will not go back into solution. But there are a lot of other proteins that will. All right, so this is critical because the ability of proteins to form these structures is what makes them have their ability to do actual work. All right. So the next biopolymer on our list is the nucleic acid biopolymer. And structurally interesting, not so much critical. The R group interaction is not kind of critical to the way proteins work. But nucleic acids are going to have some interesting things in terms of how they interact with each other. Just to flip through this slide here, we got the primary sequence, the secondary sequence, secondary structure, the tertiary structure. Now, I never, as a molecular biologist, talk about this, the quaternary structure, and that is the interaction between multiple proteins. And actually, most proteins have some kind of binding partner or somebody they're hanging with. Very few proteins work just completely by themselves. But from a DNA sequence perspective, you cannot predict quaternary structure. So consequently, I'm not going to talk about it, and I don't want to talk about it. No, it is actually another critical part, but there you go. The globin protein is held together actually as a quaternary, as a four-piece structure, and a whole bunch of different molecules that are made up of multiple proteins. So yeah, it's a critical thing. Cool. All right, nucleic acids. This is the world of DNA and RNA, and a couple of critical bits here. We've got the sugar. We have the nitrogenous base, and then we have phosphate groups. And the critical point to this is when you're thinking about RNA versus DNA, it's actually a very, very slight difference in the chemistry in that we have to have this hydroxyl group to actually link the next nucleotide. So we're going to add a nucleotide, doing a condensation reaction right there. But this hydroxyl group is not necessary for the chemistry of elongating the nucleotides. And we can, and we find organisms that have both of the OH groups. This is a ribose sugar, a 5-carbon ribose sugar, and so this is called a ribonucleic acid. If you pull one of the OHs off, or this one specifically, you have deoxygenated it, and it's now known as deoxyribonucleic acid. Really, the only difference is that the presence of just an H group here makes DNA a little bit more sensitive to acid hydrolysis. RNA is fairly resistant to acids, but DNA actually will begin to break apart if you lower the pH too much. Now, another aspect of the nucleic acid world is that DNA, because of the enzymes that break down nucleic acids, DNases are not all that tough, and they're not all that ubiquitous. But RNA is incredibly delicate. RNA degrades just if you look at it wrong. And it's one of the interesting things about the new, both the Pfizer and the Moderna vaccines, is they are RNA-based vaccines. And RNA by itself, man, is so delicate. You have to use really strong denaturing reagents to make sure you can extract the RNA without it being degraded. You keep RNA in a minus 70 freezer. RNA is so delicate. And DNA, I keep it in the refrigerator. You can keep it out on the bench. You don't have to worry about it. I mean, it'll break down eventually, but it's like way tougher. RNA is so delicate, and that's just because RNA is sort of one step away from becoming a protein, and consequently we've got a lot of checks in our world on that stuff. And so there are RNases that are on your fingertips, in the skin, on your oils in your skin. There's RNases floating around in the dust. So RNA is a really delicate molecule, and it's one of the things that is a kind of a bottleneck in the current COVID-19 testing, is that COVID-19 is an RNA-based virus. And so when they stick that swab in your nose or you spit, the virus is encoded in the protein. It's protein kind of like its coat. But then to actually get at it, they have to strip that protein away. You have straight RNA, and you then run it through some molecular processes we'll get into later. But it is pretty expensive and not just falling off a log to extract the RNA from the virus. Because if it was DNA, I mean, there are things we do sometimes upstairs where we'll just take a drop of blood or a scrape of bacteria. Don't process it, don't do nothing. Just stick it into the tube and actually I can do a DNA-based analysis on it. But with RNA stuff, man, no way. When I first saw this last March, I'm like, that's the protocol? You got to be kidding me. That's how they're going to be doing testing? And that's why testing has taken a long time to ramp up. And I'm still going, when are we going to have a stupid antibody test? Because that is so much faster. But I digress. All right, so DNA has the ability to base pair, where the adenine hydrogen bonds with thymine, cytosine hydrogen bonds with guanine. It's hydrogen bonded though, so all you have to do is heat it up and it'll break apart, the two strands. The bases themselves are linked by covalent bonds, so they don't break at all. RNA, on the other hand, is typically not double-stranded. It can form intramolecular base pairs. So you can have RNA that actually can wiggle and fold up around itself. And DNA, we draw it as a double helix, whatever. I never draw it that way when I'm trying to diagram it. But you can think of the DNA as taking a ladder and twisting it, or thinking about a spiral staircase with no handrails. And when you're walking up the spiral staircase, you're walking on the hydrogen bonds. And the actual stair sides are the phosphate sugar backbone. With RNA, though, it has the ability to fold up on itself, and you can form structures like what are known as the stem and loop structure. And this is based on the sequence. And so, in fact, tRNAs have nucleotide sequences that allow them to hybridize or base pair within to create these stems and loops. And they can fold and actually make really elaborate structures, which is a critical part of how RNA gets to work. DNA, though, typically doesn't. All right, so, now one last thing. We mentioned the idea that phosphates were critical to all this biz. Where'd my eraser go? And when we start talking about metabolism, and you probably have seen this in various places, the idea of ATP as the energy currency of the cell or whatever. ATP is a ribose sugar. It's actually part of what would be incorporated into RNA, and this is adenosine triphosphate. So we've got the base up here. We have the OH here, the OH here, and then a carbon, and then we've got the phosphate groups. I'm not drawing them right. I'm putting 4 oxygens. They may not be, and I don't care. The point being that this terminal phosphate can be popped off and used as a mobile negative charge. And so that's why ATP is said to be, the energy of the cell. It is the fact that it's a phosphate donor. And adding a phosphate, as we're about to see, can dramatically change a protein shape. And so you can have a protein that can pop and change shape, which actually is what's happening when your muscles contract. And that's because of that phosphate donor. So that's why phosphates are the energy sort of transfer system in a cell or in biochemistry, is the fact that they can transfer a negative charge. All right. So now, the next step is to think about how we can carry out chemical reactions. And this gets us into some interesting thermodynamics and biochemistry. When we think about thermodynamics, we can predict which way a chemical reaction can go. And actually, I'm going to kill this and go to the board here. We have the first and second laws of thermodynamics. The first law is stating that energy cannot be created or destroyed, which, while I'm sure critical, is not actually as important to our discussion as the second law, which states that all systems tend towards entropy or disorganization. What that means is things fall apart, but what that really means is we can predict which way a reaction is likely to go if we just let it sit. And that we're going to go from a more complex or a system with more energy, it's going to break down to a less complex system. So, just as an example, we can have glucose, and we'll throw in an oxidizer and oxygen. And this is going to break into carbon dioxide and water, simpler molecules. So we go from the more complex to the simpler. And I will just say, this is a perfect example of fire. So you can make a perfectly good fire by taking powdered sugar and sprinkling over fire, and you can make a pretty good explosion. Particularly if you add oxygen to it, you can really make yourself a really serious bomb. If we take a glucose polymer in the form of a piece of wood, you set it on fire, and it's breaking down, if it's doing, burning completely, into carbon dioxide and water. So this is a thermodynamically favorable reaction. And when we start talking metabolism, we got to talk both sides of this coin. We got the breaking down reactions, and then we had the building up reactions. And you got to balance out both of them. For our purposes right now, the breaking down reactions are a little bit easier to visualize. So if we think about a chemical reaction like fire, setting fire to a piece of wood or to a pile of coffee creamer, we can set something up like this, where we've got a graph in which the x-axis is going to be the reaction progress, and the y-axis is the relative amounts of energy. So we got high and low. And we start out with glucose up here. And we're going to end up with water down here. Now, just to make it balanced, we'll add some O2, but for our purposes, we don't have to worry about that. All right, so this is a thermodynamically favorable reaction. Now, that means that it's going to break down. And what we generate here is a release of energy in the form of heat. So this is what fire is. But I would say to you, how worried are you by having a pack of powdered sugar sitting in your kitchen? You don't consider that to be a particularly dangerous thing. It's not liable to spontaneously blow up. Even though, from the thermodynamics perspective, this is called a spontaneous reaction. I got powdered sugar in my cabinet, and I'm not particularly worried about it. And that's because there is a speed bump hump here that we don't typically diagram in this, but it's important to recognize, in that you got to actually put a little bit of energy into the system to get it to go forward. This is known as the activation energy. Or alternatively, the energy of activation. And really we can think of this as a relative stability of stuff. So stuff that is really stable, like sugar, or like wood, has got a really high energy of activation. It is going to take a very long time for it to just break down. I mean, it will. Thermodynamically, it says it will. But it's going to take a long time. Now, there are a variety of other molecules that have a really low energy of activation, meaning they're very delicate. And they can just break apart over time. There's all kinds of drugs and compounds that have to be kept refrigerated because they're labile, they're delicate. They've got a low energy of activation. I often think, everything I learned about chemistry, I learned from Bugs Bunny and Roadrunner cartoons, and I'm not sure if I say nitroglycerin, do people know what nitroglycerin is anymore? It was a classic sort of Roadrunner, Bugs Bunny thing, and that nitroglycerin is one of the components in explosives back in the day. It is very, very delicate in that it will blow up if you jar it. Now, a good, safe explosive, like plastic explosive, you can hit it, you can pinch it off, you can even set it on fire, and it won't blow up. That's a good explosive. Nitroglycerin is one that if you hit it or plink it too hard, it'll blow up. Did you say you were watching Breaking Bad?
Yeah.
Remember when Walt goes into Tuco's lair with his bag of mercury fulminate and he throws the crystal on the floor? That's an example of a chemical that has a really low energy of activation because a sudden bump will cause it to very rapidly decompose. Happily, most things don't work that way. It'd be hilarious if they did. You'd bump into something and it blows up. No, that is not the way things work. Blessedly, most things have got a really high energy of activation. And so you have to have a fair amount of heat to get these things going, or a fair amount of energy. But some things don't. Okay, so activation energy. We can think of this as striking the match. And the activation energy to get wood to combust is actually fairly high. I mean, you got to get up to about 450 degrees or so. Apparently a match is burning at 700 degrees for a few moments. It can be really, really hot. All right, so herein lies the problem. I got to break down molecules like glucose in order to get energy, and we all do. Everybody from the dog to the bacterium. The difficulty is these molecules have an inherent stability to them. Now, we can get stuff to break down by raising the temperature up And so you can think of this like we got to set up this gigantic bonfire where once we get it going, man, it's going to produce a ton of heat. But to get it going, I got to get that match, a little bit of match, a little spark. And that requires more heat than meager little biochemical organisms like ourselves can generate. I mean, I can probably run about 105. I might be able to generate 110 degrees if you really push me to it. But I can't generate anywhere close to that much energy. I mean, I just don't have it in me. I just don't work that way. Now, that's going to be a problem. Additionally, if we were to expose living organisms to a temperature hot enough to start a fire or to break down glucose, we would by definition be denaturing all the proteins required to do this. So, no, we can't run our energy systems by having to put in a lot of activation energy. What we need to do is invoke the chemical idea of a catalyst, because that's going to take care of our problems. So we shall define a catalyst speeds up chemical reactions by lowering the energy of activation. And that is our savior. That's going to make things work. If I can get energy of activations down to, for me, around 98 degrees, 37 degrees centigrade, I'm golden. I can do stuff if you can get the energy required to be around body temperature. I can actually start carrying out chemical reactions. Woohoo! And so we need to have a catalyst. And what we've got in living systems are typically, not always, but typically, protein catalysts that we call enzymes. So this is sort of a generic term, an enzyme. An enzyme is, it can be RNA, but why do we need to confuse the story? There are ribozymes, but that's not important for us. Enzymes are protein catalysts, proteins that can lower the energy of activation. And they allow chemical reactions to occur at way, way lower temperatures. Now, because we are homeotherms, we maintain our body temperature, and we take a lot of energy to do that, man. By shivering, by muscle contraction, by a variety of different techniques, we keep our body temperature at a fairly constant range. We do this because the chemical reactions in us are geared for that temperature. Now, our friends outside, plant friends and microbiology friends, don't have any way to maintain their cellular temperature. They pretty much just go whatever it is. Now, you got like lizards and frogs, snakes that can exhibit basking behavior, where they can find a patch of sun and warm themselves up. And they're doing that so they can get their body temperature up. But microorganisms and plants, fungi, these guys, they just do what they do wherever they are. What this means is when it's cold like this outside, they are going very slowly. Their metabolism is slowed way down. So they're still hanging out. They're alive, but they can't do a lot of biochemistry. They can't go very fast. So they're saying, yeah, look, I can get to that, but it's going to take me a while. I'm working as fast as I can, but it's like, walking through Jell-O. They are very, very slow in their operations. So, God, I shouldn't stand over there. I should stand in the camera. Consequently, microorganisms are going to be slowed down by reduced temperature. Probably not stopped, though. So, if we were to look at this reaction with an enzyme, we would see something that might look like this. Rather than having an energy of activation that's like that, it's now that, a lot smaller. enzyme catalyzed. And now all of a sudden, this reaction can operate at much lower temperatures. We can actually have the breakdown of glucose, and probably even happening outside, but certainly I carry it out at 98.6 degrees, man. I'm rolling. So the interesting thing is, if we raise the temperature up a little bit, all those proteins begin to lose their secondary and tertiary structure. And as we're about to see, that's what makes enzymes work. In fact, this is one of these sort of truisms that's probably a useful thing to tattoo onto yourself somewhere. And it is this, sort of a multi-layered mantra that is protein function depends on secondary and tertiary structure. And depending on what perspective we want to go, secondary and tertiary structure are dependent on the environment in which the protein is in. It's also secondary and tertiary structure depend on amino acid sequence. That means by changing an amino acid in an amino acid sequence, we can change the protein structure. And we can make a system work, maybe work better, rarely, or not work at all, by changing a single amino acid. And then the third truism would be amino acid sequence depends on the nucleotide sequence. And consequently, we have established the basis of evolution and the basis of mutation. And if you could only go back, 100 years ago, and put this on a little 3x5 note card, you'd have the Nobel Prize multiple times over. This is a critical thing to think about in terms of how the different systems and changes in the systems work. So the bottom line then is an enzyme is able to lower the energy of activation. So one last metaphor. If you haven't noticed, I'm really big on the metaphor. We're about done here. We can think of lowering the energy of activation like this. Say you get your roommate's car and you go to the top of a very high hill. And you put the car in neutral, you bring it up to the front of the hill. And your roommate obviously is worried that you're going to push his car over the cliff, because you already shocked him once. But you say, no problem. We're going to take a concrete parking barrier, one of those little concrete logs we have, and you're going to put it in front of his car. All right. That's the activation energy. That's the hump we had to get over. And now you're going to be the thermodynamics of the system, of body temperature. You're pushing on his car. You're kind of just doing this back and forth. And he's a little nervous, but he's got that big parking lot bumper in front, so he's like, okay. Now, the idea of heating it to like 450 degrees would be if you had a bunch of guys and you're just like shaking the heck out of it, blam, 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 you can get that thing to push over. But one guy, man, you can't do it. Now, an enzyme catalyzed reaction would be, rather than having a parking lot bumper that's that high, let's just put a little tiny one, like one of those little garage door stopper things, about that high in front of the car. Have it neutral, and now it's like, Oopsie, sorry. So that's what lowering the energy of activation does, is it lowers that speed bump and allows the little bit of energy that's in the system to carry this thing over. Now, this is a really great metaphor for breaking stuff apart. For bringing things together is going to require us to look a little bit more at some enzyme structures, and we will pick that up on Wednesday. But we can use the same kind of metaphorical analysis for building things up. All right, so that's all I have to say for that. No lab this week. I think we're going to be meeting up next week, but I'll let you know next week.